Hello students! Today we'll be learning all about photosynthesis and focusing in on the primary site, the leaves. As we've discussed by now, leaves are the primary site of photosynthesis, and if I was to zoom in, I would realize that they have little pores called stomata that let CO2 in. Water comes up through the stem. We're going to mix those two together along with sunlight in order to create our master product, glucose. So far, you know the basics of photosynthesis, right? Sunlight, carbon dioxide, and water combine to make glucose, what we're really looking for, and some oxygen. Today, we're going to get into the nitty-gritty of this. We're going to go into detail inside of the chloroplast, and we're going to see actually how we convert carbon dioxide and water into glucose. To do that, our notes are going to look a little different today. We are going to find a blank sheet of paper. It can be lined if you want, but white is preferable if you have it. And then you're gonna fold it in half horizontally, hamburger style or a bit like a book. When you're done, you should have something that opens and closes, something like this. And we are just gonna focus our notes on this bottom half right here for now. If you're able to print the foldable off of the hub, you can go ahead and do that and fold it in half. If not, just keep following along with this video. And then finally, one other helpful thing, you might want to grab some colored pens or pencils. Even if you don't have all these colors, just a few colors can help out with this diagram. So we're going to start off with the bottom half of our paper or our foldable. We're just going to draw ourselves the organelle where all of this takes place. And that would be the chloroplast. Remember, these are located inside of the plant cells and they're the site of photosynthesis. Sort of a round jelly bean type shape. The chloroplast is divided into two parts. If you guys remember from the diagram that we saw yesterday, he has stacks of membrane sacs, little flattened membrane sacs. They look very much like pancakes. I'm gonna make this last one maybe just a little bit thicker so we can actually see what's going on. And these guys are called the thylakoids. Thylakoids. Normally in the chloroplast we have many stacks of these, but for the sake of the diagram we'll just draw one to focus in on what happens there. The rest of the chloroplast is going to be actually filled with a kind of jelly-like substance very similar to the cytosol, and it is called the stroma. We'll have one set of reactions that take place in the thylakoids, and the second set of reactions is going to take place in the stroma. The first set of reactions take place in the light, and their main purpose is to capture the light energy from the sun. They're called the light-dependent reactions. To capture light energy from the sun, along the thylakoid membranes, we have various proteins. I'm going to make them in purple. Just several different proteins kind of chilling out here. And each of those proteins is equipped with chlorophyll, our favorite little pigment. So I'm going to draw some little green pigments on these various proteins. I'm going to remind myself that this is chlorophyll. Now what happens, remember, is that sunlight will penetrate through the membrane and it will eventually be captured right here on this protein that is containing chlorophyll. Chlorophyll will absorb the light energy and then it will treat one of our molecules, water. Now what happens is water is hanging out here in the thylakoid. The light energy absorbed by the chlorophyll is going to excite water so much that it splits apart into three different molecules. Those three molecules are going to be a hydrogen ion, so a hydrogen atom that is positively charged. The second thing he's going to split apart into is an electron. I'm going to show that as E with a negative charge. And the final thing that he's going to split apart into that kind of takes care of our hydrogen atoms, electron and a hydrogen ion, is oxygen. Now you'll probably realize that I have to split 2 water molecules to really get an oxygen, but we get the idea. And oxygen is going to then leave the chloroplast and be expelled as waste. This already takes care of 2 of our molecules in the photosynthesis reaction, right? Water is split by sunlight into electron, hydrogen ion, and oxygen waste. Now the whole point of that was to collect electrons from water. These super excited electrons that have been charged by the sun energy are going to basically bounce from protein to protein and eventually land on another special molecule. This molecule is called NADPH. Sorry, NADP. NADP plus. Okay. And he's an electron transporter. Just like the name implies, he's going to be carrying this electron around. He can easily bind to the electron, easily drop him off. He will carry him in different places throughout the chloroplast as needed. So as soon as this electron lands on NADP+, he's going to be converted into something called NADPH. Basically, this electron combines with their positive charge and becomes a hydrogen atom. Now, another cool thing is happening as we go from protein to protein. Remember, these are proteins along a membrane, so they're going to actually act like protein pumps. And each time the electron binds to a new protein, it changes its shape and helps it push through a specific ion. And in this case, what's going to happen is it's going to create a huge concentration gradient of hydrogen ions. You probably know what's going to happen next. High to low. And sure enough, this high concentration of hydrogen ions created by the splitting of water, by the electrons pumping hydrogen ions through from the stroma, they're all going to want to flow back out of the thylakoid into the stroma space. And in order to do that, they're going to flow through a very special protein channel that's called an ATP synthase. He looks a little bit like this. We'll talk about him more in class on Thursday, but for now know that as they flow down their concentration gradient from a high to a low concentration, that flow is actually generating the creation of ATP. So an ADP molecule will come up, he'll come up with a phosphate group, and as the hydrogen ions flow through, he generates ATP. Phew! Time for a quick recap. Let's take a look at this one more time. So, sunlight gets captured by chlorophyll in the thylakoids. As the sunlight is absorbed, he is going to excite a water molecule to the point where it breaks apart into 3 separate things: an electron, a hydrogen ion, and oxygen waste. The electron will bounce along the transport proteins in the membrane and eventually land on an electron transporter known as NADP+, creating NADH. As the electrons bounce along the protein transport chain, they pump hydrogen ions into the thylakoid, which will then diffuse back out through the help of a protein channel called ATP synthase, which creates ATP using that natural flow. In really brief summary, sunlight plus water is going to eventually give me NADPH and ATP. And that's part 1. The light. I'm going to put them up here. That is part 1. The light dependent reactions. Time to move on to part 2. It's going to take my 2 molecules I generated in part 1 and generate, hopefully at the end, glucose in the stroma. So let's see what happens. We're going to take the electrons from NADPH. We are going to take the ATP, also generated in the light-dependent reactions, and we're going to combine them with two other molecules. One of them is the molecule that we've been waiting on. That's CO2, or carbon dioxide. I'm going to use this little dot to indicate that there is one carbon atom. That's represented by my C here. So CO2 will come in. Okay. He's going to meet up with a special molecule called rubisco, which has 5 carbon atoms. I'm doing it in this simplified form because if I was to show you the actual rubisco molecule, it might get overwhelming. Just remember that there's 5 carbons in rubisco and 1 carbon in CO2. Now what happens is enzymes are going to help carbon dioxide, rubisco, a little bit of electron from NADPH, a little bit of phosphorus and energy from ATP. And all together, they're going to generate a new molecule. And this new molecule is called G3P, which, as you can imagine, has 3 carbon atoms. The rubisco is going to be regenerated so that he can be used again to bind CO2. And we kind of get like this little cycle going here. This cycle here is called the Calvin cycle. And it is going to be run mostly by enzymes. Let's label this. This is basically enzymes. And it is anabolic reaction. I am not going to go into all the details of these different steps because there's multiple steps involved and you don't need to know that much detail. For now, what you need to know is that carbon dioxide from the stomata, an electron that was originally from water and has now been transported over into the stroma, and then some ATP fun all combined together to spit out this molecule called G3P. Let's remind ourselves that carbon dioxide comes from the stomata, those little pores in the leaf. Man, Miss Lim, I had no idea that photosynthesis was this complicated. I know. But don't worry, we are almost there. In fact, we are just one step away from making our final product. What is our final goal product? Sugar. So you may notice that G3P has 3 carbons. If you remember, our glucose molecule has 6, right? It's C6H12O6. So that means that we don't want 3, or we don't want just 3 carbons. Instead, we want an additional 3. What that's really telling you is that the Calvin cycle is going to have to happen twice. Send us 2 G3Ps, and then we can finally make our glucose. And that's exactly what happens. So the G3P will come out, say times 2, and he actually leaves the stroma and comes out to the cytosol. You know, the jelly that kind of holds everything in your cell together. Once you've got 2 of those, you're going to have enough to finally create glucose. And reminding ourselves that glucose has 6 carbon atoms. Glucose! And we did it! So interestingly enough, glucose actually appears out in the cytosol. And this is very handy for the cell because if it's already out in the cytosol, it can easily be used all around. So if you notice, the second set of reactions that takes place in the stroma where we make glucose, it doesn't involve any sunlight. It's basically just enzymes at work combining everything to make G3P, and then G3P being combined together to make glucose. Because of this, we're going to call these reactions not the light-dependent reactions, but the light-independent reactions. They're also sometimes known as being the dark reactions, because they can take place in the dark. And that's it, guys. So we've got two types of reactions, the light-dependent that capture sunlight and release an electron to create NADPH and ATP. And then we have the light-independent or dark reactions, where we have the Calvin cycle combining CO2, rubisco, some electrons, some ATP, stirring that up with some enzymes and creating G3P. G3P is then going to leave and create glucose for us. At this point, if you've been following along with your own sheet of paper and not with the one that printed out, I want you to pause this video and go back to the hub. I want you to find the different steps that have been listed on that foldable handout, and I want you to write for yourself what those steps are. Try your best to fill in the blanks for those steps. And then if you've got questions, feel free to always come back to the Microsoft Teams call and I can help answer some of those for you. Now that you've got your foldable, I want you to actually fold it back over so that everything is hidden. And grab a pair of scissors. Now, if you remember the line that we folded down the middle here, we're going to just cut carefully along that line, only up to the midpoint. If you want to watch and see what I'm doing till the end before you do yours, that's probably a good idea. So I cut along the center of my foldable, only up to the midpoint. The reason why I'm doing this is now I can open a flap and look at just the light-dependent reactions. And then if I open up my second flap, I see my second set of reactions or the light-dependent reactions. Pretty cool. Let's go ahead and label the front for ourselves. So we've got the light-dependent reactions. And then over here we have the light-independent reactions. And if I remind myself, let's double check. So light dependent, yep, there's the sun. Light independent, yep, there's my Calvin cycle. If I remind myself, we're talking about a process that's called photo. And photo means light. And sure enough, if I look at the first half of photosynthesis, I see light being captured. And then my second portion of this process is synthesis. Photosynthesis. Synthesis means to make something. And if I look at my second half of this process, I am making glucose. Feel free to add notes, decorate, etc. however you like, but this is the basic that you should have, and it should go onto page number 31. My homework for today is to make sure that you've completed your foldable. And then you're going to take a photo of this, of the inside, please, and you're going to upload it to the hub to show me that you have watched this video and you have taken notes. I will see you guys tomorrow for some practice with these light-dependent and light-independent reactions to make sure that we really understand everything that's going on. Be safe, make good choices, and I will see you guys tomorrow.